Alrighty, good morning again. This will be the last video I give you for the next quiz. So you have a quiz coming up. I'm probably going to start it Monday. It'll be available probably for 5 days. Be like the last quiz, online. It'll just be 10 questions pulled from a random pool, 10 points. The quiz will cover the electrical characteristics of the heart, the EKGs, the arrhythmias, conduction pathway, all of that, and with a little bit of blood vessels on there as well. So this is just kind of a quick wrap up of the blood vessels. We've done arteries, all we're going to do anyway. We've done veins. Make sure you read the chapters. I believe it's 19 and 20, because again, I'm not going to be able to get enough of this crammed in these little 15-minute videos. So read the 2 chapters in Saladin. And just real quick for capillaries, we've already mentioned it. They are areas where exchange occurs. Again, that's the purpose we're moving all this blood around. So if you imagine, here's a small arteriole. Here it comes. Let's put it's arteriole. It's going to split into a capillary bed. Now I'm going to make mine pretty simple. I'm just going to put 3 little capillaries here. Of course, they'd be much more extensive than that. And of course, because exchange is occurring in the capillary bed, this is where you see that color change on your models and diagrams as oxygen leaves, assuming it's a systemic capillary bed. The blood will not be deoxygenated, and we're going to leave the capillary bed in a venule. Capillary bed. Now, as I mentioned before, I think the little video before this, capillary beds are very extensive. There's a lot of them. I mean, take a razor blade, cut yourself anywhere. You're going to bleed. Why? Because you're going to cut capillaries. You can't avoid them. Every cell in your body is within about 5 cell diameters of a capillary. So they're incredibly extensive. For the most part, they're not named. Way too many. But again, capillaries have characteristics that will maximize rates of diffusion. They do all the work of the circulatory system. So around this capillary bed, now here's where your cells would be. They're located in here, these little spaces. This is all the tissue. So these are cells all over in here. The problem is these capillaries are so extensive, you only have 5 liters of blood. If you send all that blood to all your capillaries at once, there's no blood pressure, you'll keel over dead. So you have to only send blood to the capillary beds when those cells associated with that capillary bed need it. So each arteriole feeds into a few, a half dozen or so capillary beds, which is just a series of capillaries. And again, capillaries, if you look at your slide on the PowerPoint, I'll refer to it if I remember, capillaries do not have 2 out of the 3 tunics. They have a tunica interna, or intima, which is just the endothelial layer, simple squamous epithelial basement. They got nothing else. No tunica media, no tunica externa. That's to keep them really thin. So the distance between the blood in these capillaries and the cells associated with the capillaries is really small. Shorter the distance, faster rates of diffusion. So you're going to send blood to all these capillaries, but again, you can't send the blood all at once. So right at the base of each of these capillaries, there's a little ring of smooth muscle. These are called precapillary sphincters. Now, what they do, of course, they constrict, constriction, relaxation, they call it vasomotion or vasoconstriction, vasodilation. If these little rings of smooth muscle constrict, less blood will go through that capillary. They dilate, relax, more blood will go through. So you kind of got an option here. You can think of this as pathway A, B, and C. What if these cells down here, their metabolism is higher? It's like the muscles of just this thumb bit right here, nothing else. You could constrict this sphincter, dilate that one. Most of the blood will be directed to this area. So that's kind of the idea. These precapillary sphincters, very localized control. This cell group of cells versus this group of cells. So these precapillary sphincters spend most of the time constricting and opening, They kind of flash close, flash open, flash close, flash open. And if you can watch the blood, it moves through the capillary. Here's a little blood cell there. Then it stops, moves through, stops, moves through, stops. That's because the sphincter is open, let a little blood in, then close. Also keep in mind, the diameter of these things isn't much bigger than a diameter red blood cell. They go through single file. It's very slow movement. You can watch them with a camera, you just see a stack of red blood cells. Move, stop, move, stop. Every time that sphincter closes, they'll stop. So it's intermittent flow. It's moving very slowly to allow equilibrium between the blood and the surrounding tissue to occur. So thin-walled, intermittent flow, so fairly slow movement of the blood through these capillaries. Huge surface area. I mean, if you section all your capillaries and add up all the surface area, it's massive. Again, more surface area, faster rates of diffusion. Now, these precapillary sphincters, though, they're kind of cool in that how they're controlled. Most of the other smooth muscle we've looked at in the circulatory system receives input from the sympathetic or parasympathetic nervous system. You have so many of these, you really don't have sensory and motor neurons for each of these precapillary sphincters. Your brain would have to be, twice as big. You'd look like one of those brainiac alien guys. Instead, what controls these precapillary sphincters are metabolic end products. Think about those metabolic pathways you looked at last semester. That's metabolism. What do they produce? Well, if you're producing a lot of ATP and burn it up, you produce higher temperatures. So these precapillary sphincters are sensitive to increases in temperature. What else? Well, metabolism produces CO2 and other metabolic end products like lactic acid and such, which will drop the pH. You might have things like increased CO2 levels directly. That affects pH, but it's also a chemical. You might have a reduction in oxygen levels. I'll say PO2, that's just a concentration of oxygen. Also, some other organic phosphates like DPG can be produced during metabolism. All of these things tend to cause the precapillary sphincter to dilate, to relax. That's going to send more blood to that tissue when it needs it. On the other hand, if that tissue becomes less metabolically active, it doesn't produce all of this. Temperature drops, pH rises, CO2 drops, oxygen rises. That will cause the precapillary sphincters to constrict. And when they constrict, you reduce the blood flow. So it no longer goes through that capillary bed for the moment. That way, that blood can then be sent to another capillary bed elsewhere in the body that happens to have a high demand at that moment. So they'll open up. So that's the idea. These precapillary sphincters are going to control blood flow through that capillary bed. All right, I'm going to wrap it up there. I got a couple of bits to splice onto this thing, but keep in mind, quiz soon. And also, I'm going to do one more quick lecture on blood, just its physical characteristics, hematocrit, its components, its viscosity. And then it's time for an exam. So I'm going to give you a quiz this week. It's April 5th today. And then after the quiz this week, I'm going to post an exam. It'll be similar to the quizzes, a lot more questions. You'll be able to access it through Blackboard and Connect. And then we'll be done with the circulatory system. Then I'm just going to do respiratory, digestive, and renal, and hopefully wrap up the semester. There is a lab exam coming. Wait on it. I'll have it ready before too long. Bye.